So hi everybody, and thank you for attending today's webinar hosted by Microfluidics, where we will be discussing a scalable solution for next-generation RNA technology and delivery. My name is Chris Jaquin. I'm a Senior Applications Engineer at Microfluidics, and I'll be serving as today's host, and I will also be briefly presenting. Our featured presenter today is Dr. Darrick Carter, who is the Chief Scientific Officer at HDT Bio Corp. To briefly touch on the agenda for today's presentation, I will start by giving a quick overview of Microfluidics, discussing who we are as a company, touching on some of our major applications, diving into how our technology works, and again, briefly touching on our product line as well. At this point, I'll hand the presentation over to Dr. Carter, who will get into the fun stuff, starting with the history of RNA vaccines, touching on some of the major delivery systems currently in use for RNA, discussing how self-amplifying RNAs work, going into detail on their line carrier, which is a next-generation RNA delivery system, before touching on some cost considerations for scale-up, using Microfluidizer processor technology to scale from the bench to the commercial production scale, as well as some future directions that this technology might take. Again, there will be a Q&A session at the end of the presentation, so feel free to ask any questions in the chat feature throughout the presentation, and we'll get to as many as we can at the end. So I'll start by briefly touching on who Microfluidics is as a company. So Microfluidics was founded in 1983, and we have been manufacturing high-shear fluid processors utilizing our proprietary fixed geometry interaction chamber technology ever since. We're headquartered just outside of Boston in Westwood, Massachusetts, and we do have localized support in 47 countries. To date, we have sold over 4,000 processors to over 2,000 companies. Around 10 or so years ago, we were acquired by the IDEX Corporation and have since been grouped with our sister companies Quadro Engineering, Fitzpatrick, Matcon, and Sterados to form the Materials Processing Technologies, or MPT platform. Our processors can be used at a variety of scales, ranging from R&D all the way to full-scale manufacturing, and they're used for a variety of products, including APIs, vaccines, ancient inks, coatings, nutraceuticals, cosmetics, and much more. Over our 40 years of existence, we have amassed vast knowledge of both applications and machine design, and we have robust applications, engineering, and customer support teams that are all here to help serve you should you be our customer. On this slide, I just have listed some of the major applications that we deal with at Microfluidics. Today, we'll be discussing particles similar to lipid nanoparticles, but we also work with nanoemulsions, liposomes, polymer nanoparticles, cell rupture, polysaccharide molecular weight reduction, deagglomeration, and particle size reduction as well. So if you have any questions regarding any of these applications, feel free to reach out to me, again, either in the chat feature or after the webinar. So how does a microfluidizer processor work? So to begin, you would first need to form a relatively homogeneous and stable premix before it would be introduced to the inlet reservoir. Once your product is introduced to the inlet reservoir, it is pulled into a constant pressure pumping system before being pushed through the fixed geometry interaction chamber at pressures of up to 30,000 psi. Within this fixed geometry interaction chamber, your product is subjected to very high shear rates and impact forces. And within this interaction chamber, as it is fixed geometry, all of your material will receive the exact same treatment to achieve consistent, reliable results. After passing through the interaction chamber, your product is effectively temperature controlled and then collected. So we offer two different types of interaction chambers. The first being the Y-type interaction chamber, which is shown in this top schematic here. In the Y-type interaction chamber, there are two microchannels, and after your material passes through the microchannels in two different streams, those streams then impinge upon each other in this high shear zone, which leads to intensive micromixing, which is ideal for any liquid-liquid systems. So you can think lipid nanoparticles, liposomes, nanoemulsions, applications such as those. Our Z-type interaction chamber only has a single microchannel, and the impact forces in this case come when your product hits up against its back wall. The Z-type interaction chamber is ideal for any products containing solids, so you can think suspensions, dispersions, or cell rupture. One of the major benefits of microfluidizer processor technology is that it is a completely scalable technology. And how we do so is by simply taking the single microchannel that is housed inside of the interaction chamber on the lab scale and place it in parallel on the production scale. When we do so, This ensures that so long as your formulation and processing conditions, which can include processing pressure, number of passes, and temperature, remain the same from scale to scale, that your product will experience identical uniform shear rates and impact treatment inside of each of these channels due to their fixed geometry nature. And this is how we thus guarantee linear scale-up results. Finally, I'm just going to touch on our product portfolio. So we offer a wide range of microfluidizer processors ranging from the lab to the pilot to the production scale. And we are capable of handling batch sizes as small as 1 to 2 milliliters on our smallest LV1 microfluidizer processor, up to batch sizes as large as hundreds or thousands of liters on our largest production scale machines. We also offer pharma grade or CGMP compliant microfluidizer processors on the lab, pilot, and production scale. So you can use our technology for a variety of reasons, ranging from clinical trials all the way up to full-scale drug manufacturing. So that's all the slides that I have to present on today. So I'll hand the reins over to Dr. Carter.
All right, thanks a lot, Chris. Thanks everybody for coming this morning. So as Chris alluded to, I'm going to talk a little bit about our delivery technology and maybe give you some background in RNA and RNA vaccines, because that's what our technology focuses on. And then ultimately talk about some applications and some considerations for scale up and commercial launch of these things. So, So first, I thought I'd start and dive in a little bit on the history of where RNA vaccines came from. So in 1961, the fact that mRNA was the carrier of information was discovered inside the cell. And already by 1978, people had begun working on delivering RNAs using liposomal technology. Once they had made liposomes, they then focused on actually delivering these liposomes first to human cells in cell culture, and that was around 1989. And then they developed the first RNA vaccine around 1993, and that was given then to mice. So, from this in vivo proof of concept, there was quite a lag then for first applications in humans, and it wasn't until about 20 years later in 2013 that the first human trial for an RNA vaccine began. But then, while a number of products were in development, suddenly COVID hit, and people immediately realized that this technology was a rapid response technology where you could encode the antigens very quickly and start producing them and presenting them in the right manner to the human body. And so suddenly, from first human trials to emergency use approval, only about 7 years went. And so in 2020, as you well know, the first RNA vaccines were approved. So a lot of this work was done with what's called lipid nanoparticles. And essentially, lipid nanoparticles are good technology to deliver RNA. And what they consist of is they consist of an RNA that's encapsulated inside a lipid shell. That allows for a single vial presentation, so you don't have to have a carrier and a separate RNA. However, it does require you to first produce the RNA and then at the production site co-mingle the RNA with your lipid carrier. And so each target RNA has to be encapsulated with the delivery formulation. And for a single product, that's maybe not so much a problem. But if you're going to do something like rapid responses and you'd like to stockpile your carrier, or if you wanted to go into something like personalized medicine and very rapidly make RNA and then deliver it to people, it'd be much better if you could have a bench-side type mixing technology. And so our group early on developed an early precursor to this technology called an NLC. And we've further evolved the technology since and now have a series of formulations that fall under the family of what we call the Lion technology. And what's interesting about the Lion technology is it has a very unique surface chemistry. And that surface chemistry allows you to just admix the RNA with the carrier. The RNA associates with the carrier and is then protected. With the Lion, you have optionality of placing solid particles inside the liquid core. And I'm going to talk a little bit about why you'd want to do that in some of the slides later when I talk more about how we've applied this. And this ultimately allows for enhanced stability and bioavailability. And again, I'll show you some data on that. So first, we're going to talk a little bit about the biology and about the delivery and what some of the problems with RNA vaccine delivery are and obstacles you need to overcome to make a successful vaccine. So first, we'll talk about the traditional delivery technology, the lipid nanoparticles. And basically what happens is, as you probably well know, your RNA vaccine is given into the muscle. There it is thought that it either drains to the draining lymph node or it's picked up by neutrophils and other antigen presenting sites, cells at the site of delivery. And those cells, they're recruited and then bring it into the draining lymph node where they present it. And there's where the T cell priming and B cell maturation occurs. So some features to that are that when you give the LNP, there is a drainage and there is a distribution inside the body. And you could think of that as maybe something good, but it actually causes quite a few problems with RNA delivery, and I'll talk about those next. So say you have an unmodified RNA and you're trying to deliver it with one of these lipid nanoparticles. You're really kind of between a rock and a hard place, and then that's what this cartoon is alluding to. And the reason for that is that on the one side, you need to deliver enough RNA to cause the body to start transforming and producing antibodies. But at the same time, as you present a lot of RNA, that drains to the draining lymph node, causing certain danger signals that shut off the production of the antigen. So, here's out of a press release from a company called CureVac, where they showed in their Herald study that they could go up to a 12 microgram dose, and that was probably the highest dose they could go with while not causing enough innate immune signaling. But at that dose, they only had a conversion rate of about 48% of vaccine efficacy. And so we think really what's happening is you're trying to balance giving more and more RNA with inducing more and more of an inflammatory phenotype that shuts off the production of your target antigen. And these things that you have to consider, and then I apologize for the complexity of the portion on the left, but it is a very complex signaling cascade. What happens is when the RNA gets delivered inside the cell, either through endocytosis, it's brought into an endosome, and in these endosomes, certain toll-like receptors are active, and these toll-like receptors are sensors for free RNA, and they're intended to sense viral insult into the body. And what will happen is if you have single-stranded RNA, you'll trigger TLR7 and TLR8. If you make RNA DNA complexes, you'll trigger TLR9. And if you have double-stranded RNA, you'll trigger TLR3. All of those will go through this cascade and cause the release of interferons and other kinds of cytokines. If the RNA actually escapes from the endosome and goes into the cytoplasm, it can be detected by molecules like RIG-I or MDA5. And those, again, trigger these signaling cascades that cause the release of interferons and interferon-inducible genes. Now, why is that a problem? Well, it's for two reasons. The first reason is that these interferon-inducible genes will actually shut off RNA translation. So what you see in panel A here is a standard capped and methylated RNA. Now, if you don't have that methylation, interferon-inducible, one of these interferon-inducible genes will stop the translation by binding to the 5' end of the RNA. And if you're not capped, two more inducible genes are going to bind and block translation. And the reason the body does this is because it's trying to shut off viruses that are trying to produce capsids and other kind of proteins. But if you're trying to deliver a vaccine, an RNA vaccine, you don't want this to happen because you want your RNA to be translated. And the more of this that happens, the more dose you're going to have to give. And as you do that, you're going to cause more and more side effects. Similarly, even if you have a capped and methylated RNA, and if these elongation and initiation factors start binding and try to translate it, if you have these interferon-inducible genes, they're going to bind to the elongation factors and block translation as well. So there's a number of reasons you don't want to kick off this interferon cascade. Now, companies have realized when they started developing this technology that you can't just trigger this, and one of the very important inventions was the finding that if you use pseudouridines, you can abrogate some of this TLR signaling. And you can see here in the crystal structure on the right-hand side, if you have a standard uridine, it fits quite nicely into the pocket of TLR7 and causes and thereby causes signaling. However, if you have this methyl group on the side, you can see that that methyl group collides with a portion of the TLR7, allowing for very inefficient binding and therefore not very good signaling. So adding these kind of modifications to the RNA increases the stability of your or the amount of translation that you'll get while reducing the amount of side effects. Additionally, as I referred to, if you have this capping and this methylation, you can also increase stability and increase protein translation. And by modifying the poly-A tail, you can again increase stability and protein translation. So companies that work in this space generally add most of these modifications. And companies like BioNTech or Moderna have included these in their RNA vaccines in order to reduce the amount of side effects that you see and in order to increase the amount of efficacy that they get. So, coming back to this slide, you have two options. You can try to increase your dose to increase the response, but the increased dose is going to lead to more activation of your innate pathways, and by doing so is going to increase your side effects and probably decrease the efficacy of your vaccine. So how do you get out of that? Well, there's a couple of ways to do it. One way that you could work on this is by making sure that your vaccine, when it's given, doesn't get dumped into a compartment that's very responsive and very has a high amount of innate immune cells that would then trigger these interferon productions. And one way to do that is to have your delivery system stay at the site of injection. That can add to a number of benefits. One is that you will get an increased safety profile because you won't trigger as many inflammatory cytokines, but also your vaccine isn't going to bounce throughout the body and be found inside the eye or inside the brain or produce protein inside the heart and potentially cause toxicity through these mechanisms. So it'd be very nice to have a formulation that stays very close to the site of injection. So we've gone about that and tried to develop a system where we can escape this position between a rock and a hard place. And we developed the Lion self-replicating vaccine platform. And what that consists of is the delivery technology, this Lion particle that I've mentioned earlier, that is then complexed with RNA and then delivered by an injection, an intramuscular injection to the body. When it goes there, what we've seen is it binds to the surface of cells and is engulfed in endosomes and through some pathway that we don't completely understand, the RNA escapes these endosomes and is then read by ribosomes. And what's interesting about self-replicating RNA is that in the first round of replication, it unpacks certain genes that are responsible for further transcription of the protein of interest. So in the very early times of the cell picking up your RNA, you get these cycles of translation of certain polymerase genes. These polymerase genes then start focusing on what's called the subgenomic region. And the subgenomic region is where you've cloned your gene of interest. In the case of the COVID vaccine, we've cloned the spike protein into that area. And so now the cell is focusing on producing more and more copies of the message that encode the spike protein which is then read by the ribosomes and then produced as a protein. The way it's been designed is so that it would then be exported in the Golgi apparatus to the surface of the cell and then expose the cell as a foreign protein where the immune system will start recognizing it as a vaccine. And so this kind of amplification allows you to get away with not putting in a lot of RNA into the body. And so you get we believe you get per delivered self-amplifying RNA, you get between 10 and 50 copies of your gene of interest being produced. And so if you compared that to, say, an injection of 100 micrograms of RNA, if you just had mRNA, you could potentially go down as low as 2 micrograms of RNA if you have the self-replicating system amplifying the message that you put into the body. And so by combining these two mechanisms, we think we've found a way out. By delivering the low dose of self-amplifying RNA, you don't have to increase the dose that much to get a very good immune response. And at the same time, by having this intramuscular localization, you don't trigger as many inflammatory cytokines and don't shut off the production of your gene of interest. And we've been studying this and found that yes, this is absolutely true. So if we encode the same RNA and then put it either into a lipid nanoparticle or onto the lion carrier, and then after injection, try to measure systemic cytokines, we see a profound difference between the two platforms, even though the RNA is identical. And if you're like me, and if you had your RNA vaccine, within about 24 hours, I fell asleep at my computer after my first dose at 2 p.m. in the afternoon, and that's never happened to me. I fell asleep for about 2 hours at work presumably because of these cytokines that were being produced. If we look at it in mice, we see this, within about 6 hours or so, you start seeing the production of a lot of systemic cytokines. And this goes very, very high shortly on. And of course, these systemic cytokines are early innate signals. And so if you wait a couple of days, and so at day 14, when you start getting antibody produced, you don't see any of these cytokines anymore in both systems. But we want to avoid this very early cytokine storm because it will both cause the subject, cause safety signals in the subject, as well as reduce the efficiency of your RNA delivery. And not only does it increase the safety, but we also get very effective delivery. So what you can see here is the amount of antibody being produced in a non-human primate in a monkey after injection of our RNA vaccine. And you can see that either just a prime with 250 micrograms, we were really trying to push the dose to look at the safety of the platform. And these animals were completely safe, even at doses that are incredibly high for the RNA delivery. You can see that a lot of antibodies being produced, and it's very durable. We had followed it here in this publication for about half a year, but we think that this would have continued on for a long time. And even at low doses, if you prime and boost, you get a very similar profile and get very good antibody production at very low doses. So we were quite happy with the lion delivery system. Now, I'd like to dig in a little bit deeper on what exactly the lion is. And so, as I kind of alluded before, lion in this instance is a particle that's produced with a surfactant that gives it a net positive charge on the outside, but then complexes with lipids on the inside and forms a small particle of around 50 to 60 nanometers. Inside that particle, floating in the lipid, are superparamagnetic iron oxide nanoparticles. So these small metallic spheres that I drew in there. And I'll talk a little bit more about why these are in there. But right here on the right, you can see an electron micrograph of one of these particles, and you can see the lipid portion and then this very electron dense portion of the iron oxide. And then if you put these on cells, as I kind of alluded to earlier, you can see that, that these lipid nanoparticles are binding to the surface of the cell. And then if you wait a while, they'll start disappearing from the surface. And we think they're being taken up in these endosomes and then delivering the RNA to the interior of the cell. So one thing we really like about the line is that it's easily manufacturable and very scalable. So if you want to include iron oxide nanoparticles, you take these particles and you coat them. You first clean them, then you coat them with the oil. As Chris alluded to, prior to feeding it into the microfluidizer, you'd like to have a very homogeneous feed stream. And so what we do is we use a high-speed homogenizer first and homogenize the particles. Then we feed it into a microfluidizer and do high-pressure homogenization, which breaks these particles down. And ultimately, they come to rest at an energetically favorable size. And in this case, our size is between 50 and 70 nanometers. So then if we take these line particles and then we complex them with RNA, we can start studying the biophysical properties of the complex and the ability of the complex to prevent the RNA from degrading. And here on the bottom left, what you see is there's a slight size shift, as you would expect, as the RNA complexes to the line particle. And once it's complexed, you can even store the RNA for a short period of time at room temperature, even in liquid form, which we were quite impressed with. And then you don't see any growth of these particles over time. They're quite solid. But then if you challenge them with RNases and you compare that to naked RNA, what you can see is that naked RNA is very quickly destroyed by RNase and completely destroyed within hours. If you add the lion particle, it completely protects the RNA from RNase digestion. We see a little bit more faint bands because of the complexing, and then it's very difficult to get the RNA back off in the lab, but we really don't see much of a change over time. And so now getting back to some of the advantages of line and some of our thoughts about how this line is working. One thing is that we think the RNA, and I kind of alluded to it as kind of a submarine-like structure. We think the RNA is not actually inside the particle like it is in an LNP. We think it's more like a semi-submerged submarine, that it's sitting at the surface interacting with the positive charge. But it can't be completely surface exposed, otherwise the RNases could get to it and start clipping it. So we think it kind of is somewhere in between there. And that has some advantages because what happens when you complex something like a string with a rubber ball? Well, if you look at the total free energy of the system, what you're doing is, and you assume that the free energy of the system is staying the same, what you're doing is you're shifting the entropic component, so the flexibility, the amount of waving around that the string can do, you're trading that in for an enthalpic component, so the string is actually bound. So that binding energy is traded for an entropic energy. Now, if you look at the Gibbs free energy equation, the entropic energy is dependent on temperature. And so if you increase the temperature, you'll very quickly increase the entropy of the system. And so the less entropy in that system that you can have, the less temperature dependent your system is going to be. And presumably you're going to be able to make a more thermostable vaccine that you can ship, instead of at -80, maybe you can ship it at 4 degrees in a refrigerated system, which is much easier to deal with in terms of logistics. Another advantage is that these iron oxide nanoparticles, of course, they're super paramagnetic, so they have a magnetic resonance signal. And when we had first started the company was going to focus on cancer because COVID hadn't been invented yet. And so what we were going to do is we're going to be able to follow the delivery of our innate agonists or our cancer vaccines into the body and see where they distribute within the body. And here's just an image of a muscle where the particle or the vaccine has been injected into an animal, and then we put it into an MRI, and we're able to see very nice MRI signal with the vaccine being delivered. And so, this is of course very exciting because we think we can follow the vaccine as it goes through the body. So we can both do localization studies, but for our cancer applications, we can also follow where this is going and see if it's actually localizing towards tumors in our preclinical models. So that's a little bit of background about challenges of RNA delivery and the lion particle and what we feel is some of the advantages that our system has. And so now I'm going to shift gears a little bit and talk about scalability. So I like to talk about S-adenosylmethionine. It's a cofactor. It's important for many different biochemical reactions, including RNA production. But I worked with this as a kid in a undergrad lab, and we bought quantities of like 250 microliters, which is maybe a very large droplet on your faucet or so, and it costs about $205 if we wanted to do a couple of reactions of SAM, as we affectionately called it. Now, if we did these calculations and said, hey, we want to make RNA at commercial scale, how much SAM would we need to buy? Well, it becomes quite a daunting amount. Now you're talking 4,400 liters. So it's almost a swimming pool, a small swimming pool full of S-adenosylmethionine. And that translates into 4 kilograms of this chemical. And if there was no advantages of scale, you'd have to pay $3.6 billion just for the single cofactor. And then this isn't looking at all the other components. And so, one of the things that I highly encourage people is when they're looking at the components that they want to combine when they make something like a nanoparticle like the lion, is to look at what is the effect going to be as you go to commercial scale, because you're not just going to be in the lab, you're going to go to the bench side, from the bench side to the bedside, and from the bedside you're going to go to commercialization. And once you get to that scale, are you going to be able to find enough of these raw materials in order to satisfy your commercial market, assuming that in the best case that your product is approved. And it's not just cost of goods, but it's also, how are you going to go from the lab scale of manufacturing ultimately to commercial scale of manufacturing? And I'm visualizing this here with these little coffee machines. And so, the lab scale, you see this little cup full of oil, and this is a French press, a small coffee maker that you might use at home to make 1 or 2 cups of coffee. And here, if you use something like a Microfluidics M110P machine for microfluidization, you'd be running at about 100 mils per minute. So, you could make a reasonable amount of formulation, but you probably wouldn't be thinking about commercial scale here at all. You'd be thinking about personalized medicine or phase 1 trials or so. Now, if you wanted to start doing tech transfer, you might want to think about a more serious machine, something a little bit more automated, like this coffee maker shown here, that can make a little larger quantities, but maybe something a little bit more reflective of what you might, of how you might do this for pre-commercialization scale. And thinking again about microfluidics machines, you'd be thinking here at a level about the 110 EH. Where you can flow at 200 mils per minute, and you can make reasonable quantities here. You definitely could satisfy phase 1, phase 2 clinical trials with the EH and produce good amounts of formulation for this. Now, if you want to start getting serious and start getting pre-commercial and then start doing early production, or have something that models your commercial production for phase 3 clinical trials, you're going to start having to get something a little bit more robust, something that can flow at least a liter per minute. And thinking about microfluidics here again, we'd be thinking here about something like the M815 or machine, where you can flow at 1 liter per minute. It's good for early commercialization. Maybe thinking about 1 million doses or so per production run. And then finally, if you want to get really serious, you're going to have to buy one of the commercial machines, and this would be something like the M700, where you can start flowing at 4 liters per minute, and now you can start thinking about making tens of millions of doses in a single run, and as you keep the machine running, or if you do it in a flow-through manner, you could satisfy the world demand for a certain vaccine with a machine like that. And so the nice thing about working together with somebody like Microfluidics is that you're really integrated from the early lab stage all the way through commercialization, where you can make 1,000 to 2,000 vials with your early stage 110P, like the one we have in the lab. Then you go into your GMP facility and start making 5,000 to 7,000 vials for phase 2 trials, or maybe early phase 3 trials if you're thinking that way. And then going into 1 million doses per year. And for a cancer vaccine or something like that, that might be the scale you want to be at. You go to the M815, and then finally, as you go to full commercial scale and try to make 100 million doses per year, you go to something like the M700. And so we've transferred our technology. A nice thing about the line that I alluded to is that the manufacturing is fairly straightforward. So we've been transferring it as part of our mission to enable other countries, especially emerging economies, to produce commercial scale. We've transferred technology to India, Brazil, China, Korea. So we're transferring the technology throughout the world for production. And we were quite satisfied to hear that in India, this technology has gotten emergency use approval. So it's now the third mRNA vaccine, I think, that's been approved in the world. And it's the first one that got full approval for, as a self-replicating RNA. And we think that's related to the safety profile. So if you look at the amount of side effects that you see in these early clinical trials, you can see a great reduction in reported systemic and local effects after injection. And we think that's directly related to some of the advantages of the lion carrier. And then if you look at the efficacy, we see very good efficacy. We see neutralizing titers, that are on the scale of what these other RNA companies are seeing. So we're very happy of that, of escaping that rock in the hard place and producing something that's both safe, able to reduce the amount of RNA that you need, but still get very good vaccine outcomes. And so we're looking forward to more approvals in other countries as we move through the regulatory process in these countries. But, as I alluded to earlier, we initially intended to use this in a cancer setting, and this story here is to me is one of the great stories of somebody who had metastases throughout her body. And you can look here in the liver, you can see these dark spots that are highlighted with these yellow arrows. They were metastasized from a breast tumor that this patient had, and they basically told her that she had months to live, a couple of months to live. And she went in and was treated with TILs that were reactive against four of the neoantigens that she had. And she had a complete response, and then all these tumors kind of melted away. And so what we'd really like to do is to use our technology in order to be able to produce these neoantigens in a very efficient manner and induce cellular responses that would then allow the patient to reject these tumors and hopefully, produce outcomes like this in a number of different patients with a very cost-effective delivery system. And so what you could envision in having something like this is both, if a new virus emerges, or you have a patient who has a tumor and you want to do personalized medicine, you would take that tumor and go through sequencing the message repertoire inside this person and looking for neoantigens or other conserved antigens that you might be able to give them. You would then transmit that to a production facility that can make the RNA. They would then send you the RNA. and then you would complex it with the line at the bedside, inject it in the patient, and potentially within a week you could have you could have material for treating a patient in a commercial in an immune therapeutic setting. Now, we have a great proof of concept of that in that, when COVID came out, we had a DNA synthesizer in the lab. And so I believe we might be the first people in the world who actually had a vaccine in animals, because it took us a week to have the first mice immunized. And if you see the date of publication of our non-human primate paper, we were the first to show efficacy of a COVID vaccine in non-human primates. So we're quite proud of that. And so we think that this system that we designed for cancer immune therapy can be used in pandemic and emerging virus settings as well. And then finally, I kind of alluded to this early on, but the line also enables you to do something like lyophilization, and with smaller RNAs, we see even in liquid, we see very good stability. However, if you freeze the entire formulation, so the line with the RNA inside a vial, and then you remove the water in a vacuum in a lyophilizer, you can form very nicely formed cakes. And these cakes very quickly dissolve when you add water. And so what you could envision is you can ship these freeze-dried vials of vaccine either at 4 degrees or at room temperature, potentially, to the patients or to the pharmacies. And this becomes very important in developing economies where you don't necessarily have -80. And even in the US, we've had quite a bit of problems dealing with the -80 cold chain. And so this allows you to get out of that -80 cold chain. And we've seen great stability with the line at 2 to 8 degrees. We think we can get that to years with the RNA once we have freeze-dried formulations. So we're definitely working on that as one of our future directions. And so, yeah, I think, that's pretty much all I had. Hand it back to Chris.
Yeah, thank you so much, Dr. Carter. It looks like we have around 10 to 15 minutes for questions, should you have any. So again, feel free to ask those in the chat feature, and we can get to as many as possible. It looks like we have one to start out with. The first one being, could you please comment on the size distribution, PDI, and zeta potential of the line delivery system? And I think we can go back to a slide discussing that.
Yeah, so thank you. Yeah, whoops.
Sorry, we had it. Okay.
Yeah, go back to that one. Because you can see here the size distribution curve on the bottom left. And as I alluded to, most of the particles are around 60 nanometers, and they kind of range from 50 to 70 nanometers in our size distribution. Our PDIs are generally pretty good. They're pretty much they're quite a bit below 0.2, which is kind of our cutoff for people who aren't familiar with particle sizing or dynamic light scattering, which is how this is determined. The size distribution kind of is an indicator of where the particles are. Like most things in nature, they form a Gaussian distribution, and that Gaussian distribution is centered around a maximum likelihood size, and that maximum likelihood size in our case is 60 nanometers, but it goes from 50 to 70. The PDI is the polydispersity index, and that shows you that's an indicator of how variable your particle distribution is. And so generally you want a PDI below 0.2, and we easily get into that. And the zeta potential is the surface potential of the particles, is the average surface potential, and that's usually expressed in millivolts. And I believe ours is around 15 plus 15 millivolts, so it's positively charged, and that changes as you add RNA, and some of that charge is then neutralized by the addition of the RNA. So I hope, Francisco, I hope that answers your question there.
Perfect. I believe that's the only question I see at the moment. So again, feel free to ask them in the chat feature. Okay, looks like we have another one coming in. Asking, so after adding water to the cake, could you de-agglomerate the particles using ultrasound?
You don't need to. After after you add the water, the particles actually go back to the pretty similar to the polydispersity and the particle distribution that we see when we first produce them. So you don't need to deaggregate them. They form a nice solution right after you add the water back to it. And of course, we want that to be a feature because, you know, if you want to make this easy to administer in the pharmacy, you don't want them to have to go through another step prior to injection. You just want them to add water, pull it up in a syringe, and then inject it into the patient. I think I see another question.
Yep, so it looks like we have another one. So yeah, this question is around whether ultrasound or high shear might damage the RNA structure.
Excuse me. Yeah, that's a great question. And yes, you could damage, especially the RNA, especially long RNAs, by adding any kind of force to them. You know, they are like a string that can break. But you know, what I like about the question is it comes back to something I didn't touch on in the production of LNPs, and that is if you have very long RNAs and you have to put the RNAs through a high shear mixer or a high shearing process like a microfluidizer, you can destroy these large RNAs, and so you do start seeing what we call frazzled or ragged ends of the RNA after you put it through. So that's another way that by not having to put your active ingredient through the microfluidizer, by just being able to bedside mix it, you also protect the integrity of the RNA.
Awesome. It looks like we have another couple of questions here. So the first one being, is the authorized vaccine in India lyophilized?
Yes, it's being distributed that way.
Perfect. And then the second question being, are there any results that show how long the dose is effective? How long after the initial dose is a boost required?
That's a great question, but we don't have the clinical data yet. We only have the non-human primate data that seem to indicate that we might not need a boost for a year. And we relate that to two factors. One is the ability of the self-amplifying RNA to really give you a good durable signal of antigen. And then the second is that we're starting to see a lot about T cell immunity after self-replicating RNA administration. And so we think that might increase the ability to produce long-lived plasma cells or memory cells. So, you know, unfortunately, I can't give you a good answer in people yet because our molecule hasn't been out there long enough to study how long this duration might be. But certainly in non-human primates, it looks like we'd probably be at least a year out, and hopefully, you know, 2 to 3 years. It'd be great if we don't need a booster constantly.
Awesome. The next question I think I might be able to handle, asking what shear rate is ideal for good stability. From the perspective of microfluidics, at least, and feel free to kind of add on at the end here, Dr. Carter. When you're adjusting the shear rates, it's very formulation dependent. So essentially on a microfluidizer processor, you would choose a specific interaction chamber and choose a specific processing pressure that kind of work in conjunction to provide a specific shear rate to your particle. So it is very formulation dependent, but really I guess the major goal that we typically look at is particle size and zeta potential. And those are really the two functions that you will look at when it comes to the stability of the particle. And it is very formulation dependent as to what shear rate you might need to apply to your system in order to achieve the desired particle size and zeta potential. Is there anything you might add to that, Dr. Carter?
No, I think that's great response.
Okay, perfect. The next question we have here is someone asking for some clarification. So, do they understand correctly that the paramagnetic iron nanoparticles are actually purely for diagnostic purposes in cancer therapy, but have no essential function in the COVID-19 vaccine formulations?
Well, so there's two things. We're not 100% sure yet. But as you could envision, with my thermodynamic explanation there, if you have a solid particle, you might actually increase the stability of the formulation so that a vaccine formulation could be more stable in the presence of the iron particle. Not exactly sure about that one. We're still studying that. But, iron oxide and iron oxide nanoparticles also have a number of other very interesting properties and interact with the immune system. And you can probably look in the literature about how they're being used. And so we are pursuing that quite a bit because we think that there are functions that are encoded in the iron oxide particle that may go well beyond the diagnostic purposes in cancer therapy. But certainly it's a really cute application to have this ability to follow things with MRI. I really loved that image that I showed you when we first saw it.
Absolutely, that's very interesting. The next question is around the use of mRNA. So, can this be equally effective to deliver mRNA? Excuse me. So RNA that's not self-amplifying.
Yes, absolutely. And we've looked at a number of different types of RNA, and so I think our platform initially was designed, and our chief technology officer, he was from AlphaVax and was one of the first to make a self-amplifying RNA. And so, of course, we have a lot of expertise in that area, but we have shown delivery of mRNAs and as well as RNAs that signal through the innate immune system. So if you're thinking about immune therapy or actually treating people who have been infected with pandemic viruses, you can see that triggering the innate immune system can be used as treatment. And so we have made lions that are in that area. And so as I kind of alluded to earlier, the lion is kind of a family of formulations. And we think depending on the application, we would make different types of lions. But in a simple response, the lion that I showed you can deliver mRNAs. That's absolutely true.
Awesome. It looks like we have a question kind of building off what you just kind of alluded to there, where you can make many different formulations with the Lion platform. The question is, how are formulations screened and optimized?
It depends on what we're trying to do. We do use a lot of design of experiment software so that we can talk about multiple variables. We have replaced the core with different types of cores, so we've gone away from just iron oxide to other inorganic oxides and things like that. We can also change the type of oils that are inside, and then we can change the ratios of both the detergents that form the exterior core, as well as the ratios of the detergent to the RNA, which is what we call the N to P ratio or nitrogen to phosphate ratio. And as you can see, that opens up an incredible high dimensional space where it's very difficult to test every single formulation. So what we do is do the design of experiments, and then we test formulations that look promising in animals and look at their ability to produce whatever effect we're trying to get. So as I alluded to, if we're trying to give an oral formulation, that's going to be tested very differently than we're trying to optimize an intramuscular formulation. And so the readouts are quite variable. But I think, changing different variables within the formulation, looking at their stability, their ability to complex RNA, and then their ability to have an in vivo effect is how we screen these formulations.
Perfect. And it looks like we have a couple more questions. We'll probably make these the last two questions. So the first is for Dr. Carter. So by any chance, are other materials like silica or nanocomposites other than iron used? They're interested in learning more.
Yes, we have tried other things, and there are applications that you can envision. So one that I like is with aluminum nanoparticles, where we can get positron emission and look at PET signals. So you could use it for kind of diagnostic purposes. Or you can envision something like alum, where you have another adjuvant added to your RNA vaccine that might enhance your immune responses. So yeah, it's a fun thing to do in the lab, is to use other materials to make lion-like particles, and you can, and as you do, they change some of their characteristics, which is kind of cool.
Awesome, thank you so much. The last question I think I can handle. It's asking, is there a limit to the size of the nanoparticles using microfluidizer for a given formulation? For example, will it not decrease below 100 nanometers even after increasing the shear force? And I will say, for a given formulation, there's a few processing parameters you can look at. Typically, if you are trying to get as small as possible, we would recommend using the full processing pressure of 30,000 psi. And if you are processing your sample through a given interaction chamber at a given pressure, you can run it through for a number of passes. And typically, we will see for most formulations, after a certain number of passes, you will start to see the particle size kind of plateau. So, for example, you might reach 100 nanometers after the third pass, and after the fourth and the fifth pass, you're still seeing it at 100 nanometers. So the important thing to note there is that while you can vary some of the processing pressures or the processing parameters in our technology, that is really only kind of one side of the equation. The other side is obviously the formulation, which is just as, if not more important, when it comes to achieving a proper particle size. So I hope that answers that question. We have also been getting the question, will this recording be shared? And we just wanted to mention that yes, we will make this recording available via our website after the fact, and we will contact all of the attendees with information as to how to access the presentation. So again, I just wanted to thank Dr. Carter for joining us today and giving that very interesting presentation. And with that, I believe we will conclude the webinar. Thanks everybody for attending.
All right, thank you.